Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 2. This is lecture number 13. The topic is exceptions of Mendelian genetics. We have already had one lecture on this topic where we discussed a few examples, and in this lecture we will further explore examples that violates the principles of typical Mendelian genetics. Discussing agenda for the topic will be extending Mendelian genetics for two or more genes, epistasis, polygenic inheritance, nature and nurture, the environmental impact on phenotype. Lecture outcome. After watching and listening this lecture, students will know about different genetic crosses where the Mendelian laws are violated. Now, extending Mendelian genetics for two or more genes. In our previous lecture, we discussed different dominance relationships like incomplete dominance and codominance. Then we discussed multiple alleles and pleiotropy. All have to do with the effects of the alleles of a single gene. Now we consider two situations in which two or more genes are involved in determining a particular phenotype. In the first case, we discuss epistasis, which is the phenomena in which one gene affects the phenotype of another gene because the two gene products interact with each other. In the second case, called polygenic inheritance, multiple genes independently affect a single trait. So what is epistasis? Epistasis is a phenomena in which a gene at one locus controls the expression of another gene at a different locus. And then we will discuss the polygenic inheritance, which is a phenomena in which a single trait is controlled by more than one gene. So such traits which are controlled by more than one genes are called polygenic traits. Eye color and skin color are the examples. Now we discuss epistasis. So in epistasis, which is derived from a Greek word meaning standing upon, it refers to the fact that in this phenomena, a gene at one locus controls the expression of another gene at a different locus. The phenotypic expression of a gene at one locus alters that of a gene at a second locus. Now we will discuss one of the examples where you will see this phenomena. In Labrador Retrievers, which is one of the breeds of the dogs, and they are commonly called as Labs, the black coat color is dominant to brown. So the coat color is either black or brown, and the black is dominant over the brown. So it means that if only one allele for determining black color is present, then it will control the phenotype. And for the phenotype to become brown, both the alleles should be in recessive form. So let's designate the capital B to represent the allele which determines black color, and the small b to represent the allele which determines the brown color. Now for a lab to have brown fur, which is a recessive trait, the genotype must be BB. Both the alleles must be in homozygous form, the recessive alleles, only then the genotype, the phenotype will be brown. Now these dogs are called chocolate labs, but the story is not so simple here. There is one more angle to this story, one more twist in the story. Now this is for the gene which determines the fur color, which exists in two alleles, capital B and small b. Now a second gene which is present at some other locus, it determines whether or not pigment will be deposited in the hair. So just recalling the typical Mendelian genetics, a capital B which is dominant controls the black fur color, and a small b which is recessive controls the brown fur color. Now they must be doing their jobs, but they on their own cannot determine the phenotype. They are under the effect of a gene which is present at some other locus. So a gene which is present at some other locus determines whether or not pigment will be deposited in the hair or not. That is, whether black pigment will be deposited or whether brown pigment will be deposited, irrespective of the phenotype, irrespective of the genotype at that locus. So this is the example of epistasis. We further explore it. Now the dominant allele. If the allele at that second locus is dominant, then what will happen? The allele which are present at the first locus, they will express themselves. If it is capital B, the color will be black. If it is small b, the color will be brown. But it will only happen if the allele at the second locus is dominant, and if the allele at the second locus is recessive, then the dogs will have yellow fur color, and the dogs will not exhibit the phenotype controlled by the alleles at the first locus. So you can see here, if the allele at the second locus is dominant, then the phenotype will be controlled by the allele at the first locus. So BB is controlling black. Similarly here, black is dominant, so it is heterozygous dominant. So you should also see the allelic combination at the second locus. Now come here, the allele at the second locus is recessive. So if the allele at the second locus is recessive, it will automatically determine yellow fur color, irrespective of what allele combination is present at the first locus. So you can see here, the allele at the second locus is recessive, so the dog will have yellow fur color, irrespective of what allele combination is present here. Now here also, the alleles at the second locus are recessive, so it will automatically determine yellow color, irrespective of what allele combination is present at the first locus. And if the allele at the second locus is dominant, then what allele combination is present at the first locus will be determined. Now here it is dominant, so the phenotype will be controlled by the alleles at the first locus. It is heterozygous dominant, so the dog will be black. It is homozygous recessive, and the allele at the second locus is also dominant, so the phenotype will be controlled by the alleles at the first locus. Now see here, the allele at the second locus are recessive, so the phenotype is yellow irrespective of what is present here. This doesn't mean anything. If the allele at the second locus is recessive, if the alleles at the second locus are recessive, then yellow color will be determined. It doesn't mean anything then. It will only have the alleles at this locus will only have their effect if the alleles at the second locus is dominant. But if the lab is homozygous recessive for the second locus, that is E, then it will automatically mean yellow fur color irrespective of what is present at the first locus. So in this case, the gene for pigment deposition, this is said to be epistatic, or it is controlling. The gene at this locus is controlling the expression of the genes at this locus. Similarly, the genes at this locus are controlling the expression of the genes at this locus. Now here, as you can see, the black, the dominant allele determines black fur color. Here both the alleles are dominant. So the fur color of the dog must be black, but it doesn't happen because the alleles at this locus are under the effect of the genes which is present somewhere else. So this is epistasis. So we can say that the alleles here are controlling the expression of the alleles at this locus. Now what happens if we mate black labs that are heterozygous for both genes? So we mate the black labs that are heterozygous for both the gene loci. So the outcome of this cross will be, you can see here, so because it is independent assortment, so all types of allele combination is present in sperms, and all types of allele combination is present in the eggs. So then you construct a simple Punnett square, and you will find that the 9 of the dogs are having black fur color, 3 of them are having chocolate, and 4 of them are having yellow. You can also see the genotype of both the allele loci. I hope now you understand this concept quite well. So this will be the outcome of this cross. If two dogs which are heterozygous for both allele loci are crossed, remember this is because of independent assortment where all type of allele combination in the gametes is possible. Now polygenic inheritance. So polygenic inheritance is a phenomena in which a single character is controlled by more than one gene, and polygenic characters are of course very hard to treat through gene therapy, because you just cannot carry out gene therapy for polygenic traits. You cannot just incorporate lot of genes into a patient's system. So the conditions caused by polygenic polygenes are difficult to treat. Now, Mendel studied characters that could be classified on an either or basis. That is, only two variations in the phenotype are possible when you observe flower color. That is, in Mendel's case, flower color was either purple or white, flower position axial or terminal, plant height tall or dwarf, seed color yellow or green, seed shape wrinkled or round. There were no intermediates. There were only two distinct phenotypic expressions of a trait. But many characters such as human skin color and height are not one of two discrete characters, but there is instead a continuous gradation. For example, when you observe the height of individuals in a population, you will find a lot of values. There will be no two extremes. Some people will be 5 feet, some will be 5 feet 2 inches, 3 inches, 5 feet 4 inches. So a whole lot of variations in the height will be there. And why? Because these characters which does not show two extremes, which show lot of variations, are controlled by polygenes. Now these are quantitative traits, that is, they can be measured. Quantitative variations usually indicates polygenic inheritance, that is, that they are controlled by many genes. An additive effect of two or more genes on a single phenotypic character. Height is a single phenotypic character and it is controlled by more than one genes. So therefore you see a whole lot of variations. Similarly, the skin color, the eye color, you see lot of variations. It is because these characters are controlled by more than one gene. Now in 2014, a genomic study of over 250,000 people found almost 700 genetic variations associated with over 180 genes that affect height. So there are over 180 genes which control only a single character, which is height, and therefore there were 700 genetic variations. Now many variations were in or near genes involved in biochemical pathways affecting growth of the skeleton. So variations in genes means the changes in the nucleotide sequences, and as a result they vary from each other and control the trait to a different extent. And just for your knowledge, a new allele arises through gene duplication events. Skin pigmentation, which is yet another example, is also controlled by many separately inherited genes. Now we will talk about nature versus nurture, whether the phenotype is wholly and solely controlled by the underlying genotype, or the underlying genotype is also under the effect of environment. So this is yet another deviation or violation or exception of typical Mendelian genetics, where the information in the genes will only be expressed in a particular way under a particular set of environmental conditions. For example, a person has genes which determines height, but the nutritious quality of this person is not good. So despite the presence of the genes which determines tallness, the person's height will not increase because the genes are not in a suitable environment to express themselves. Similarly, you may have observed the leaves of the plants. Now all the leaves of the plants have the same genome, but some of the leaves are smaller in size, others are larger in size. Why? The leaves which are exposed to the sunlight, they will be larger in size because they will have more opportunity to carry out the process of photosynthesis and prepare food for themselves. And the leaves which are away from the sunlight, there the process of photosynthesis will take place to a lower level, and therefore their size will become small. So these are examples of the effect of the environmental conditions on genes. What I have explained already, you can see here from these lines, the color, the shape of the leaves, and the height or the exercise help in building the muscles. The sunlight helps in darkening the skin, and the experience improves performance. So these are some of the examples where the genes are being influenced by the environmental factors. Similarly, identical twins who are genetic clones, they may accumulate phenotypic differences if they are exposed to different environmental conditions. There may be differences in their phenotype, in their physical outlook, in their cognitive level, depending upon the experiences they face. So this is yet another example of violation from typical Mendelian genetics. Whether human characters are more influenced by genes or the environment in everyday terms, nature versus nurture, is a debate that we will not attempt to settle. So you can also search this debate on the internet. This debate started back in 1960s, where some of the people were of the opinion that it is the nature, it is the innate message in the genes which is expressed, which is advocated throughout the life of an individual. On the other hand, other people were of the opinion that the early life experiences have a profound effect on the behavior of individuals. So you can also refer to the internet for further inquiry about this. We can say, however, that a genotype generally is not associated with a rigidly defined phenotype, but rather with a range of phenotypic possibilities due to environmental influences. So the genotype controls the phenotype, but to varying degrees, depending upon the environmental conditions. For some characters, such as the ABO blood group system, the phenotypic range has no breadth whatsoever. That is, a given genotype mandates a very specific phenotype. There are some traits which are controlled in a pretty straightforward manner, just like the Mendelian genetics, where there is no room for variations, and whatever message is there in the genes is expressed in the phenotype. And there are other characters like the RBC count and the WBC count. They do vary a bit between individuals with altitude, with nutritious level, with exposure to infectious agents, with physical activity. So there are variations in these parameters. Generally, the phenotypic range is broadest for polygenic characters. So if a character is controlled by more than one genes, then there will be several phenotypic variations for such characters. Example being the skin color, the eye color, the height, etc., where a lot of variations are exhibited by the individuals in the population. So environment contributes to the quantitative nature of these characters, as we have seen in the continuous variation of skin color. So here also environment do have impact even on the polygenic traits, where the skin color, although it is controlled by many genes, but still the variation in the skin color do occur as a result of environmental conditions. For example, the people living in areas where there is extreme heat, they do develop tan coloration as compared to the people who live in areas where the temperature is low throughout the year. So geneticists refer to such characters as multifactorial. Which type of characters? The characters which show a lot of variations, and the characters which are controlled by many genes, meaning that many factors, both genetic and environmental, collectively influence phenotype. I hope you have understood the lecture. Thank you very much. Allah Hafiz.